All right. Hi, everyone. My name is Dr. Nicole Ackermans, and I'm going to talk about the correlation of dietary proxies used to measure tooth wear in a long-term feeding experiment in sheep. This is basically the conclusion of my entire PhD work while I was at the University of Zurich. If you have any questions, please @ me on Twitter at AckermansNicole. So, tooth wear can be used to reconstruct diets of extinct herbivores. And by using different proxies to look at the state of their teeth, we can create an estimate of their diet and paleo environment. This is because the amount of abrasives in their diets from either tough or soft plants, which are phytoliths, or grits, which come from open or closed environments, they all wear the teeth differently. So this allows us to have sort of an idea of where they used to live. So different proxies are used to detect wear at different scales. First of all, using the naked eye or at low magnification, we can observe macroscopic wear on the macroscopic scale. And we use this technique called mesowear to do this. And this technique consists in basically looking at the shape of the cusps, whether they're pointy or not, and the depth of the valley between the cusps, highlighted here in red. And this signal basically develops over years and years and represents the individual's general lifetime diet. But if we move to a much higher magnification, we can distinguish microscopic wear looking at the individual facets of the tooth, as here with some putty. And this wear only develops over a few days or weeks and then gets overwritten constantly. And this is often why it's called the Last Supper proxy. So these two techniques can be used, but within microscopic wear, there are different techniques as well. First of all, one called micro wear, where you look at scratches and pits left on the enamel surface, or another technique called dental microtexture analysis, and that's the one that we're going to be focusing on during this talk. And this is what a DMTA analysis would look like here. So here's my question to you. If you're a paleontologist and you have a reduced set of teeth, you're obviously going to want to perform as many analyses as possible. But this is because you want to understand these diets at various scales. But what happens when these two proxies show two different diets that don't correspond? This is a problem that we often run into in the literature. You will have a mesowear signal that's showing you an overall grazer signal over the animal's lifetime. But at the same time, you'll have a microtexture signal that's telling you browser. So you can either assume, well, I guess this animal just has a mixed diet, or you can assume this animal just ate browse right before it died. The problem is we don't know. We cannot know for sure because we don't know how microscopic wear gets transformed or develops into macroscopic wear. What happens when there's a microscopic scratch on the tooth surface? Is it pushing the tooth material to the side, or is it removing material? And if that's the case, if you get an accumulation of microscopic scratches, will this eventually result in a worn down crown? We don't know. So how do you figure out if microtexture and mesowear are even measuring the same thing? Well, you do a large feeding experiment where you feed animals different diets and record the development of wear over time. But how do you perform a large-scale feeding experiment? You hire a PhD student. That's me. So we performed this long-term experiment on sheep using different experimental diets of varying abrasiveness. We had a controlled diet with no abrasives, and then 6 other diets increasing in concentration of abrasives as well as size. And we fed this to 7 groups of sheep over about a year and a half. During this time, we applied multiple proxies to the sheep's teeth in order to understand the development of wear over time. And we did this using CT scans at multiple moments during the experiment. So ideally, if microtexture and mesowear are measuring the same process, then we would expect the same signal across the different proxies. So we have already published the results of each of the proxies separately. First of all, here mesowear. And what we found is that the mesowear signal barely changed over the entire experiment. And so this led us to believe that you know, even if they ate very, very intensely abrasive material, they didn't change too much. So mesowear must be more of a lifelong dietary general signal rather than a seasonal one, as we thought before. We also applied this technique called absolute wear. Basically, we're measuring the entire tooth volume as well as the volume of the crown segment and the root segment and seeing how it changes over time. What we noticed is that the crown segment was reducing in volume, but interestingly, the root segment also increased in volume. So we suspect a sort of compensation mechanism where something like cementum is being opposed to the roots to compensate for the wear on the crown. Finally, we also applied dental microtexture analysis to the teeth. And with our different types of abrasives, we noticed that the smaller sort of dust-like abrasives created a polishing effect, while the larger abrasives had an increase, they showed an increase in microscopic wear. So now that we have all of these analyses separately, our aim is to compare them and see if indeed there are correlations between these different proxies, since they were all applied to the same animals over the same period of time. Before I do get to the results, though, I want to explain a little bit what dental microtexture analysis looks like, DMTA for short. So as you can see, it has a lot of complicated parameters. There are 57 in total that are regrouped into different functional groups. So this is a technique that was adapted from the field of material engineering, which is incredibly ingenious, but some of the parameters are redundant. They sort of measure a similar thing. And so these different functional groups help us identify the topography of the tooth. You have the volume of the different scratches and topography, or the height and things like that. But possibly this could be simplified. And also, so this is another great picture of our experimental sheep, just to note that we use the M2 as the tooth of reference, the upper right M2, as this is the only tooth where all three parameters were applied. So what do we get? This is the correlation matrix of all of the DMTA parameters, correlated to each other on the upper right M2. It looks complicated, it's not. It's pretty simple. Blue is the positive correlations, red is the negative, and the larger the dots, the stronger the correlations. As you can see, for a lot of the groups, the parameters are hardly correlated to each other. For example, with the height, or here with the volume, very high correlations. Additionally, a lot of the parameters are correlated to the other groups. So this indicates a lot of redundancy and would justify the use of a reduced data set to sort of simplify this technique and make it easier to understand. Quickly, if we just look at the same sort of plot that we applied to each of the teeth where we did the mesowear, so the maxillary M1, M2, M3, and the mandibular M2, it kind of looks like I copy-pasted the same plot 4 times, but actually they're just incredibly similar, which was a little bit surprising. You'd expect maybe a gradient of the M1 having a different kind of wear than the others, but they're very similar, which was interesting. So if we break down the parameter set and we just take one parameter per functional group, we get this little square here, which is much more manageable. And as you can see, there is still a high amount of correlation happening. It also makes it easier to compare this to the other parameters from the other kinds of proxies. So I made a little orange square here for the mesowear proxies and a red square for the absolute wear proxies on the right side, if you're colorblind. And well, there's some interesting things happening. First of all, the mesowear, the different types of mesowear do correlate with each other, but this is to be expected because they're just different subsets measuring a similar thing. And in the literature, it is known that cusp shape will correlate with occlusal relief. Interestingly, they didn't correlate at all with the volume changes, although you'd expect if the crown is being worn down for the mesowear to change. And one of the theories is that it could be that the crown is losing volume, but the shape is staying the same, and therefore the mesowear would not change. only some of the DMTA correlated to mesowear and absolute wear, but much, much less than you would have expected. So this was the most interesting finding for us. And if I just take two examples, this is kind of the scatter plots that we would get for the correlations. The mesowear one looks a little bit weird because it is just these measures. We just have 1, 0, or -1 for the type of crown shape. But still, you get a strong correlation. And the thing is, you know, this is what you would want to see, but you'd want to see many, many more of these. In this instance, this is the occlusal relief correlated with the slope of the features on the tooth, and this one is the difference in crown volume with the area of the features on the tooth. So if I may conclude, first of all, overall, as it usually happens, we have much, much more to learn about how microscopic wear becomes macroscopic wear. And this is essential if we want to make these conclusions where we're determining the diet of the animal based on these two parameters together. We need to understand how they work together. And this will also help as we finesse the DMTA analysis procedure between labs, between machines. As you see here, I put the one that we used. And so overall, more work on this will help everyone get on the same page and help us all understand better what is happening. One way to also get a better grip on this would be to use in vitro experiments. First of all, they look really silly, which is interesting, but mostly you can make as many as you want, add different quantities of grit, different sizes, different types phytoliths, and every time apply microwear, mesowear, and see how things evolve. Or see how the microwear would eventually become mesowear over without having to do an entire feeding experiment and wait a year. You could just speed up the chewing and see what happens. Finally, there might be differences between enamel hardness or softness. When someone else from our group, my colleague, Dr. Martin, applied the same techniques and the same diets to rabbits, she found many more correlations between the proxies on this animal, which has softer enamel. Similar things happen when in publications where the studies are looking at cartilage and another where they're looking at steel, many more correlations happen in the cartilage study, and we think there might be something happening where that the softer materials are more prone to sort of better correlating, at least with DMTA. Again, this is to be explored, and it's only a hypothesis. So overall, we need to work together to achieve a better understanding of these processes. So thanks for listening. Please join the Q&A Tuesday, October 13th at 12 p.m. so that I can answer all of your questions. And if you forget, you can always @ me on Twitter at Ackermans Nicole. Thank you and enjoy the rest of your conference.